Namaskar. Today we'll study the life cycle of virus. Virus is an ultra microscopic infectious parasite. It depends on a host cell. Why it depends on a host cell to survive? Because it lacks protein synthetic machinery. There is no ribosome. It has a nucleic acid core which it has to transfer, and then it it will depend on the host cell ribosome for its protein synthesis. So it has a host dependent mechanism of multiplication, and since it depends on a host cell, this virus enters a host cell, so we cannot differentiate between the cell metabolism and the virus multiplication. So these are the challenges for treatment of virus. Then this virus, once it multiplies inside a host cell, then the host cell bursts and the virus is released, and in that case only after the release, when they infect fresh cells, the symptoms appear. That means when the symptoms appear, already the virus are multiplying and replicating, so it is difficult to diagnose. Means when we diagnose, by the time it is already infected, already late. And when we select a drug for this virus, it also affects the host cell. So it is equally toxic to the virus as well as the host cell. So these are some of the challenges of virus. And if we see the structure of a virus, it consists of three parts. One is the envelope, the outer envelope, lipoprotein envelope, on which on its surface we have the glycoproteins. Then inside the envelope there is a layer of capsid which consists of capsomeres. So this is one capsomere, this is one capsomere, and this layer is called capsid. And inside the capsid we have a nucleic acid core which may be either DNA or RNA. So this nucleic acid core and capsid together is called nucleocapsid. And when this nucleic acid, when we say this nucleic acid core is there, it can be either DNA or RNA. And accordingly, the virus can be of two types, DNA virus and RNA virus. So the virus which contains only DNA is DNA virus. The virus which contains only RNA is RNA virus. Examples of DNA virus are pox virus, papilloma virus, parvovirus, adenovirus. And examples of RNA virus, rhabdovirus, rotavirus, picornavirus, that is polio virus, influenza virus, a type of orthomyxovirus, paramyxovirus, retrovirus, example HIV, human immunodeficiency virus. So these are some of the examples of RNA virus. And retrovirus or HIV is a type of RNA virus, but it is something different because it has one enzyme called reverse transcriptase, which so we know the transcription is a process in which the DNA, the double stranded DNA, is converted to single strand RNA. So first the double strand DNA is converted to two single strand DNA, single strand DNA, and then each single strand DNA then one can synthesize, that means from one DNA we can have two DNA, and the enzyme here is DNA polymerase, and out of these two strands, one strand is called the sense strand and the other is called antisense strand, and the antisense strand of DNA helps to synthesize your messenger RNA, and the enzyme here is DNA dependent RNA polymerase, DNA dependent RNA polymerase. So this process is called transcription, DNA to RNA, and reverse transcription, that is RNA to DNA, and that reverse transcription is required in this retrovirus or HIV. That's why the reverse transcription is required. So this virus is called retrovirus. So let us see the life cycle of a virus. A virus first attach to the cell surface, then it absorb and penetrate into the cell. Then there will be uncoating of the virus, and after uncoating, the nucleic acid core will be released, either DNA or RNA. If it is a DNA virus, it will release the DNA. If it is RNA virus, it will release the RNA. More DNA will be formed by the action of DNA polymerase. DNA to messenger RNA will be synthesized by RNA polymerase. Similarly, RNA virus will provide viral RNA. By the action of RNA polymerase, more RNA will be formed. So this messenger RNA then binds to the host cell ribosome. So now the host cell action starts. Messenger RNA binds to the host cell ribosome. There will be synthesis of the viral protein, and in some virus there is action of protease, so that these viral protein which are synthesized will be converted to structural and functional proteins. Then all of them will assemble together. There will be an encapsulation or encapsulation, and a daughter virus will be formed, and then the cell will burst and the virus will the mature virus will released. In case of H1N1 virus, that is swine flu virus, there is one enzyme called neuraminidase which helps in budding and release of the virus. And this protease is mainly present in HIV and also present in hepatitis virus. So when antiviral drugs are to be given, we can target all these steps. We can target the DNA synthesis or RNA synthesis, or this attachment or adsorption, penetration or uncoating, or even assembly or release, or this protease. All these can be the targets. Then if we see the retrovirus, so one type of RNA virus is retrovirus, which contains reverse transcription. So that RNA has to be synthesized to DNA first. So let us see the example of HIV, human immunodeficiency virus. This is a type of retrovirus, and the HIV, if you see the structure of this HIV, you can see on the envelope there is glycoproteins, the glycoprotein 41, above the glyco-GP41 there is glycoprotein 120, and inside there is not only the nucleic acid core, that is the RNA, but it also contains the enzyme reverse transcriptase, it also contains protease, and it also contains integrase. So these are the enzymes present in HIV. So what happens here also, the attachment occurs when this GP120 binds to the CCAR5 chemokine receptor 5, and the GP41 binds to the CD4 receptor. So CD4 receptor and CCR5 or CXCR4 are the co-receptors here. So virus first attach to it, then it will penetrate into the cell. Here the cell is the T lymphocyte. HIV affects the T lymphocytes. Then there will be uncoating, and the RNA will be released. So this RNA, and there will be also release of the enzymes. So first there will be action of the reverse transcriptase. So this is viral RNA. Then by the viral reverse transcriptase, there will be formation of viral DNA, and this viral DNA then it will integrate with the host cell DNA, and we call that as provirus, and here the enzyme is integrase enzyme. Then this provirus, from this by the action of host cell DNA dependent RNA polymerase, there will be synthesis of messenger RNA, and this messenger RNA now will bind to the host cell ribosome. There will be protein synthesis by the action of protease. There will be structural and functional proteins, and this proviral DNA, some genomic RNA also be synthesized, and all together they will assemble and a new virion will be there, and the cell will burst and this virion or virus will be released. So this is all about the life cycle of virus, and here also we can target HIV by drugs like which can inhibit this attachment. Then reverse transcriptase can be one target, integrase can be another target, the protease can be another target. So this is all about the life cycle of virus. Thank you.